Vanakkam. Welcome to this video on classics in neuroscience. We have been looking at signaling molecules. In this set of videos, we have been looking at the first growth factor. In this video, we will be looking at how connections are organized. In the previous video, we saw an organizer principle. Remember who suggested this organizer principle? Who is the proponent of this idea? Mangold, is it not? Mangold and Spemann. From the previous couple of videos, this is Mangold and Spemann. But here we are looking at organizing the connections, because when we discuss the organizer principle, we ask the question, what organizes the organizer? That is the question that we attempt to tackle here. How the connections themselves are organized, that is the question that we will attempt to tackle in today's class. We saw in the previous videos that NGF, or the nerve growth factor is crucial for survival of the cell and growth of the cell. A question is, what are the factors that govern or determine the specific connections that one neuron makes with a target cell, with another neuron? What are the factors that control what kind of connections are made by a given neuron with the target neuron? Because this is not controlled by NGF, perhaps, is it not? Because NGF only makes sure that a given cell survives when compared with other cells in the competition of other cells. We also saw that there can be natural cell death, that is pruning also happens. This is what we saw in the previous set of videos, but here we are interested in identifying what factors determine the kind of connections or which connections happen with a target cell. Also, what are going to be the target cells and how this happens, that is an orderly arrangement of neurons that target an orderly set of neurons. That is an orderly arrangement of cells or neurons in one region, region 1, leads to orderly arrangement in region 2. Because we observe this kind of orderly arrangement in the central nervous system, so we need to identify how this orderly arrangement or specificity of connections is happening. This is crucial to improve our understanding of how these connections happen. Cahal suggested that when axons grow out to make connections, some sort of chemical interactions are involved. This was the original suggestion made by Ramon Cajal. This was followed up by John Langley, who performed experiments to check if this is true. So, these experiments were performed in the 1890s. So, John Langley, what he did was he cut the preganglionic fibers in the superior cervical ganglion and observed how reinnervation was happening. Reinnervation is this process in which there is restoration of dead cells. So, when you cut, these neurons go back and try to identify their target cells and again connect, make new connections after the cut. So, he observed that the reinnervation happened in a precise manner. So, he cut and he activated specific preganglionic neurons, and he found appropriate or exact responses that were found before the cut. So, the response that was found before the cut was present even after the cut. That means what? That means that reinnervation happened in a very specific manner, that this regrowth of this neuron happened in a manner that was very specific. So, each neuron knew where the target cell was, somehow it knew where the target cell was and attached it to that, or as in synapsed with that target cell. So, this is stunning to think that this could happen in this way. This led him to speculate. I am going to read from the exact text from the material, Langley's material. There is some special chemical relation between each class of nerve fiber and each class of nerve cell, which induces each fiber to grow towards a cell of its own class and there to form its terminal branches. At bottom then the phenomenon would be a chemiotactic one. This idea came to be called as the idea of chemiotaxis. Late, remember, this was the late 1800s or late 1890s or early 1900s, a good half a century before the identification of NGF itself. Okay. So, this essentially anticipated NGF and the concepts of NGF a good half a century before it was properly discussed. Then this idea was pursued by Paul Weiss in the 1920s. He trained in Vienna, then worked at Chicago and at Rockefeller University. He performed experiments by grafting limbs or grafting certain body parts and observing muscle contraction in the original body part and in the grafted part of the body. What he observed was that there was muscle contraction and coordination that happened along with the exact body part that was grafted. So, the original contraction and the grafted limb were contracting together. So, this is what he observed. So, Paul Weiss speculated that innervation of motor neurons happened in a rather random fashion, and specificity of activity or specificity of activation happened due to the specificity of or the patterns of activity So, that is, it is not the spatial distribution of the cells that mattered, but rather how in time the activity varied that matters. This temporal changes in activation controlled how variation happened in space. This was the idea. That is, the comparison was with a musical instrument, say for example a violin. When you move a particular string, you are going to hear a particular tone, right. Likewise, the output is a specific activation pattern that is activated, or this specific tone is activated by a temporal pattern, is what was suggested by Paul Weiss. Because this involved this music related idea, this came to be called as the resonance hypothesis. But then there were some questions, because during muscle activation all the neurons were not simultaneously activated, but rather only the active muscles were being activated. That was the discrepancy, right. That is, only those muscles that need to be activated were activated, not all of them were activated, because resonance theory posited that the activation pattern is what governed which particular limb or which particular action is going to happen. But what was observed was that only specific neurons were getting activated. So, this meant that the resonance theory was being heavily questioned or almost on the verge of being disproved. But if you look back in retrospect, this is the idea that a spatial distribution of activation can happen due to temporal distribution of neuronal activity. This idea was controversial at the time because it was way ahead of its time. Today, this is a accepted fact. Today, we accept that a particular spatial activation can happen because of changes in temporal activation. This is an accepted principle today. Paul Weiss continued this line of questions, and he found that in some culture systems, growth appeared to follow physical features of the culture dish. For example, scratches in the culture dish were followed in the growing specimens. So, he suggested that axons were bundled together and they stayed together throughout the trajectory till they reached their targets. So, there was really no need for any kind of chemical signaling to happen. That is, they start at a point and they travel together. These axons, these bundles of neurons travel together until they reach their targets. So, the trajectory remained the same. They are in constant physical touch. They are bundled together and they are in physical contact with each other. That by itself led to this theory being called as the contact guidance theory. This suggested that there was no need for any chemical signaling to happen between the neurons. These ideas of Paul Weiss were followed up by one of Paul Weiss's students. Roger Sperry was one of Paul Weiss's student. He did his PhD with Weiss and went on to do postdoc with Carl Lashley. Carl Lashley was a behavioral psychologist who was a strong believer in this idea that there was no discrete actions or discrete regions in the brain or in the cerebral cortex. The brain as a whole acted without having a need for specific regions of the brain to be active for specific activities in the body. The brain or the cortex as a whole was active to produce specific actions. This was Carl Lashley's idea. Roger Sperry studied with Weiss and Carl Lashley. Both of them are opponents of this idea of specificity, because Paul Weiss suggested that there was no need for this chemical signaling to be present, and Carl Lashley suggested that there was this no presence of any kind of discrete actions happening or discrete regions in the cortex responsible for some discrete actions. But Roger Sperry decided to perform his own experiments where he studied the connections from retina to optic tectum. Retina to optic tectum. He studied this in newts, goldfish and frogs. These had excellent capacity for regeneration. So, these formed excellent model systems. Remember our early discussions about the need for identification of a proper model system for addressing a specific question. In the first week, we saw there is a need for us to identify a good model system to address a specific question. So, Roger Sperry's model system was newts, goldfish and frogs, specifically the pathway between the retina and the optic tectum or the retinotectal pathway. So, the exact experiment that Roger Sperry performed was this. He transected the retinotectal pathway, the pathway that goes from the retina to the optic tectum in these animals such as newts, goldfish and frogs, and then he rotated the eye by 180 degrees. This is crucial. He rotated the eye by 180 degrees. So, first is transection of the retinotectal pathway. Second is rotation of the eye by 180 degrees. Remarkably, after the transection, these neurons went on to make re-innervations with their original targets. The original map of the retina onto the optic tectum was preserved, despite the fact that there were two insults to the system. One is transection, the other is the rotation of the eye by 180 degrees. So, the original map was preserved, but remember the eye is rotated 180 degrees. So, because the eye was rotated 180 degrees, the animals behaved as if they were upside down. This is unique because this showed finally that there is indeed specificity, that specific regions in the retina connected with specific regions in the optic tectum that was found even after transections, that is even during re-innervation you are going to find this specificity. So, these animals behaved as if they were upside down because the eye is rotated 180 degrees. So, that is the key intervention where he rotated the eye by 180 degrees. This caused the animals to behave as if they were upside down. They believed that they were upside down. This is the key intervention that made sure that there was this specificity. This led Roger Sperry to postulate or to speculate that there existed some sort of biochemical identification tags between axons and their targets that were preserved even after transection, that were preserved during re-innervation. So, he extended this idea by saying that these are not only preserved during re-innervation, these are present during original development itself. That is, there is this specificity of axons to specific targets, specific regions, for example, in this case the optic tectum, that identify each other using some sort of biochemical sensor, biochemical tag, some identification tag used to be there. It has to be there for this to happen. This idea came to be called as the chemo affinity hypothesis, that there was some sort of biochemical affinity between the neuron that is attempting to reach and the neuron that is the target. There was some sort of affinity with each other. These ideas were published in the 1950s, specifically 1959. Remember, such an idea was earlier proposed by John Langley about half a century earlier. So, these ideas of chemo affinity essentially challenged the original theories of Paul Weiss and Carl Lashley, who were Roger Sperry's advisors in PhD and postdoc. they provided evidence to dismiss the notions that there was no specificity, because these two professors, these two teachers were among the leaders in the field. But here comes one of their own students and challenges their idea and provides evidence, convincing evidence, to suggest that their ideas were not entirely correct, actually were quite wrong. So, Victor Hamburger suggested, later recounted this particular results, saying that I know of nobody else who has dismissed their PhD advisors and postdoc advisors works at the same time like Roger Sperry or something of that sort. This is the meaning of what he said, but not necessarily the specific words. The specific words are something like, I know of nobody else who has disposed of the cherished ideas of both his doctoral and postdoctoral sponsor, both at the time acknowledged as leaders in their fields. It is difficult to challenge the leaders, especially if you are their student. But this is how science grows. Remember our discussions in the first week. You need to have this boldness to question and a proper convincing set of results. He had a great model system. So, this model system of using the retinotactile pathways in these animals became the norm for the next few decades, for example, in this line of study. So, the retinotactile pathway proved to be an excellent model for studying this idea of chemoaffinity. Studies that followed the original study by Roger Sperry used this model system and assumed some variation of the original chemoaffinity hypothesis. But the question remained as to what are the biochemical identification tags that Roger Sperry discussed about. This was not found until much later in the 1980s and the 1990s, when cell surface identification molecules were identified or found, only much later. Remember, Sperry's experiments were in the 1950s, the late 1950s. Although the specific cell surface identification molecules were found later, the theory that there are these biochemical identification tags came in the 1950s. So, this idea preceded the experiments, or this idea preceded experimental evidence that came much later, 2-3 decades later, for example. But that is regarding the identification tags. What about arrangement? Because in the original Sperry theory, specific regions in the retina were mapped onto specific regions in the optic tectum. So, there was a map with xy coordinates, for example, something of this sort, maybe not exactly this. I have just drawn a grid. There has to be a map or some sort of a map that must be present in the optic tectum for the information that is received from the retina. How is this happening? Sperry postulated that there were spatial gradients that identified specific x and y coordinate in this map, and perhaps there were some genetic mechanisms. There were some genetic mechanisms behind specific synaptic connection patterns that happened in this system. But later it was shown that there were indeed multiple signaling molecules that controlled how cells or neurons interacted from development to repair. There were many signaling molecules that were involved. So, this research, these results came much later. So far what has been identified, the interest in the nerve growth factor expanded into identifying specific cell signaling molecules that controlled or somehow enabled cell to cell interactions. So far what has been identified are growth factors and those that identify a group of cells. But there has been considerable challenge in identifying cell specific or individual cell level molecules. That there has been considerable amount of challenge in this. However, there are some examples where some specific molecules guide axons to different glomerular targets. For example, in the olfactory system, there is some identification of individual cell level or cell specific molecules in some cases, but not everywhere. So, with this we come to the end of this video. In this video we looked at how connections were being organized. We started our discussion from chemotaxis, then resonance theory, contact guidance, or rather chemiotaxis, then some form of chemotaxis, chemotaxic affinity, just suggested by Sperry. With this we come to the end of this video. Thank you very much for your attention.